I'd like to introduce our speaker today, Dr. Billur Akkaya. She got her MD at Hacettepe University. Sorry for the mispronunciation.
Hacettepe, that's fine. Totally
Okay,
fine.
thank you. And her PhD at University of Oxford, UK. She did postdoctoral studies at NIH, and she was a member of the Laboratory of Immune System Biology, NIAID. Her research interests are multiple sclerosis and neuroimmunology, and she studies the mechanisms of Tregs, or regulatory T cells, mediated immune suppression in the context of autoimmunity and cancer using state-of-the-art techniques. Her lab is currently funded by the NIAID New Innovators Award, DP2, for exploring new precision therapies for autoimmune diseases and solid tumors. And today she's here with us from the Ohio State University to talk about deciphering the specificity and molecular mechanisms of Tregs using novel approaches. So welcome. Thank you so much for joining us.
Thank you very much for having me. And thanks very much, Monica, for this generous introduction. So as Monica stated, I'm a new investigator. I started my lab in 2021, in the second half of 2021. In Ohio State University, and I'm an assistant professor here, and I will tell you about how we try to decipher the specificity and molecular mechanisms of Tregs using novel approaches. All right, so let me give you a quick overview about what I am going to talk about. So in the first part, I will tell you about the mechanisms of Tregs. So what we know, what we don't know, and what we would like to know about how Tregs work. And I will continue by telling you about our efforts in terms of delving into molecular mechanisms of antigen-specific Tregs in mice and human. And in the second part, I will switch gears a little bit. This will be more of a work in progress section of my studies. We are still developing all these tools, but I'm happy to share with you our progress so far. And okay, so regulatory T cells constitute 5 to 10% of CD4 positive T cells in the peripheral blood, and these are marked by the expression of the transcription factor FOXP3. So the FOXP3 locus is something good to know, and it is pretty clear-cut in mouse systems in terms of identifying Tregs. I won't get into the details of how unreliable it might be in human systems, but in mice, we can study mouse immune system and mouse Tregs by altering FOXP3 locus in mice. Okay, so here is a dramatic view about what happens if a mouse, an adult mouse, loses all its Tregs. So this is a special mouse, FOXP3 DTR. So there's a diphtheria toxin receptor knocked into this FOXP3 locus. And what happens is that when you treat the mice with diphtheria toxin, the mice just, their Tregs die. So the comparison is the wild type mouse. So if you follow this mouse, you see that within 20 days, all these mice are succumbed to severe autoimmunity and they die. So this tells us how dramatic it might be to lose all our Tregs all of a sudden. So despite it is a very small population, it is important to understand how they work. And so far, we know that there are antigen-specific and bystander suppression mechanisms by which regulatory T cells work. During my postdoctoral studies, I showed that regulatory T cells can steal peptide MHC2 complexes on dendritic cells with a fine specificity, and they lead to a pinpoint specificity inhibition in the T cell responses. So here I am just trying to depict everything with color coding. So this is a Treg that has a red TCR, and it this Treg steals these red peptide MHC2 complexes, and this results in this T cell with a red TCR to get a minuscule stimulus compared to another T cell with an irrelevant TCR. So this is one of the mechanisms by which regulatory T cells work. And there are also numerous bystander mechanisms, such as downregulation of CD8086, IL-2 consumption, which results in a lack of survival in the T cells in the vicinity. And Tregs can secrete immunosuppressive cytokines such as IL-10, TGF-beta, IL-35, and this list goes on forever. And we still don't know how exactly in different contexts Tregs suppress immune responses in vivo. Probably a combination of all these mechanisms, depending on the context, but it is fair to say these mechanisms are not mutually exclusive to one another. I will tell you a little bit more about my own interest, about the antigen-specific suppression. So we did a key experiment, and I like to show this experiment to make the point. So we created a setting where all T cells were subjected to the same bystander effects. So bystander effects are, the inflammatory mediators or whatever you can think of on the bystander list. So what we did was we obtained antigen-specific TCR transgenic T cells from some donor mice. So these are OT2 specific T cells that recognize this peptide, OVA peptide, and SMARTA cells, S cells, that recognize LCMV-associated peptide. So the question was, when we give a mixture of different specificity naive T cells into this mouse and create an immune response in this mouse by injecting the mouse with another set of dendritic cells. So these are freshly isolated dendritic cells, antigen presenting cells, obtained from a normal mouse. And we preload them with two different antigens that are recognized by each of these T cells. So the question is, when the dendritic cells have these two antigens simultaneously presented on their surface, would you expect antigen-specific Tregs suppress both T cells, or one T cell but not the other T cell? And we got the answer within 3 days of, transferring all these cells. So this is showing you an overview of how the cells proliferate. So basically what we do is we label these cells with a proliferation dye. And as the cells divide, this dye is diluted into daughter cells. And this is an indication of proliferation. And by measuring the percentage of these dye dilution, you can calculate how much these cells proliferate, or this is an indication of their stimulation. Okay, so the upper row shows the proliferation status of O T cells, and the bottom row is ST cells. And in the control mice, we only have the dendritic cells and the T cells, no Tregs. So in the mice that received all Tregs, what we saw was quite dramatic. Although these Tregs were able to bind the dendritic cell and get activated, they did not inhibit ST cells, but they only preferred to inhibit OT cells. So there was a complete antigen match. And vice versa, in S-Treg transferred mice, OT cells were perfectly normal and happily dividing, and ST cells this time were inhibited. So this was black and white antigen specific, and this was in contrary to the previous observations that were done in vitro. So I always tell my students, well, it is really important to do in vivo experiments that back up the concepts and results you obtain from in vitro experiments, and this is one of the examples of that. And I am skipping a lot of things in between, and I want to share with you what we found at the bottom of this interesting antigen-specific suppression. Here we use another set of antigen-specific TCR transgenic system, and I'll tell you why. So these are this time HT-regs that recognize HEL antigen, and this is an MT-reg that recognizes multi-cytochrome C. So what we saw was when we measured specific peptide MHC2 complexes, not the total MHC2, but specific peptide MHC2, that means that presents a specific antigen. In this case, it is multi-cytochrome C. We saw that Tregs specifically take on peptide MHC2 complexes that they recognize. Even though dendritic cell presents two antigens at the same time, and Tregs are able to bind dendritic cell surface. And this is the control cell type here. They don't acquire, so this is gating on the Tregs. They don't acquire MCC complexes, whereas MTregs acquire MCC complexes, and vice versa is also true. And looking on the dendritic cell surface, what we saw was quite dramatic as well. So the double pulse DC, so the DC that has both antigens, is the dashed histogram, and this is measuring the peptide MHC2 complexes again with this antibody. And after dendritic cells contact with these Tregs, one or the other, we saw that HTregs did not diminish these complexes on the dendritic cells. However, MTregs did. So there was a dose-dependent reduction on the peptide MHC2 complexes on the dendritic cells, and this was very much antigen specific. And this is still, how this is happening is still puzzling us. And we also asked the question whether this also happens in the normal conventional T cell synapses. And I'll just show you pretty pictures again to make the point about the differences between regulatory T cells and naive and activated T cells. So we decided to look at the interaction. So because we saw something different happening in the interaction of antigen-specific Tregs with the dendritic cells, we decided, okay, let's focus on these interactions early on. So we immobilized dendritic cells loaded with antigen on silicon chips. So these are fibronectin-coated silicon chips, and just co-cultured cells, just to let them interact for up to 3 hours. And then we visualize them by scanning electron microscopy, confocal microscopy, and also we did flow cytometry. What we saw was quite dramatic, and we started to get clues simply by looking at how these cells interact. So this is an interaction between naive T cell and the dendritic cell, and you see that the Treg has a round morphology and picture perfect synapse interaction. And activated T cells, they got bigger, they preserved their round morphology, and you would see more prominent microvilli, but you would still see the cells are interacting in a way similar to the naive T cells. However, when you look at the dendritic cell Treg interaction, you would notice that Tregs have a different morphological appearance, and they look more like amoeba than just round T cell T lymphocytes, and they have very long philopodia that they extend to the synaptic space. And if you, as much as we could resolve into these membrane bridges between these cells, we noticed that the naive T cell dendritic cell interaction harbored rather narrow and multiple membrane bridges. And we know that these bridges contain the TCR and their co-stimulatory receptors. And activated T cells presented a more mature synapse structure with considerably wider nanodomains, membrane nanodomains. And looking at Tregs, what we saw was there was a bigger membrane interaction, which was suggestive of an even more mature synapse structure compared to activated Tregs that carry the same TCR. And we were curious that whether these morphological adaptations may underlie peptide MHC2 uptake, and we resolved into this time frame by electron microscopy and followed our peptide MHC2 movement in these naive T cell, activated T cell, and iTreg. And within this time frame, what we saw was this is the dendritic cell sort of full of endosomes that are carrying peptide MHC2 complexes that are marked with quantum dots. And this is a Treg that is currently in synapse with the dendritic cell, and you can appreciate that there are some early endosomal structures that also harbor these peptide MHC2 complexes. And let me tell you, Tregs or any mouse T cells, they don't have MHC2 or peptide MHC2 to start with. So it has to come from somewhere else. And this is, in this case, coming from the dendritic cell. And we were also we were able to detect some isolated patches of peptide MHC2 complexes on the surface of Tregs as well. And this is just a quantification. Within this time frame, you would see more quantum dots indicative of peptide MHC2 complexes accumulating inside and on the surface of dendritic cells. And this is just a total MHC2 patch that ends up on the Treg. And okay, it is tricky business to study these antigen-specific Treg interactions, and you have to resort to several tools to be able to do this. So we have done this quite a lot in mice so far, and the idea is you would, use a TCR transgenic FOXP3 reporter mouse strain and either isolate these cells out or differentiate your naive T cells into induced Tregs ex vivo. However, I would like to focus on the ways to derive Tregs and conventional T cells, antigen-specific Tregs and conventional T cells out of human PBMCs. And right now we are trying to develop we are optimizing our ability to transduce the TCRs of interest into the T conventional cells and Tregs that are derived from a normal human. And so I didn't want to reinvent the wheel. I found a great collaborator, Todd Brusco. Dr. Brusco is the director of Diabetes Research Institute from University of Florida. And I approached him to ask for his expertise and his help in developing lentivirally transduced Tregs. So they call these Tregs Treg avatars, and I just wanted to do the experiment of peptide MHC2 capture in this well-controlled human system by his help. And so what we did was basically this was done by Todd's lab, and they are still helping us with deriving all these TCR transduced Tregs and helper T cells. And now my people, as my lab gets more experience, we have also started to do this on our own. So anyway, so he gave me the Tregs that carry TCRs of known affinity and known specificity. So these Tregs and T helper cells were transduced with an antigen that's glutamic acid decarboxylase antigen is specific peptide residue, and these were formerly derived. These TCRs were formerly derived from patients, type 1 diabetes patients, and we know that they recognize this peptide sequence. And he also provided us HLA-DR4 risk allele carrying type 1 diabetes patient PBMCs, from which we isolated B cells and monocytes as a source of antigen presenting cells. So this is rather a proof of principle experiment where we try to understand whether human Tregs also have the capability of stealing antigen from antigen presenting cells. And what we saw, and this is preliminary data, and we are currently repeating all these experiments, again, with the same set of TCRs, but different samples. What we saw was regardless of the affinity, so this is a low affinity TCR, this is a high affinity TCR, and this is the amount of peptide MHC2 captured by Tregs versus effector T cells. So red histograms show the Tregs, black histograms are the effector T cells. And you can appreciate that from B cells, there are heaps of antigen that is stolen by regulatory T cells that have the high affinity TCR. But this doesn't mean that the low affinity TCR Tregs don't also steal peptide MHC2. They also do, and I would like to say that regardless of the affinity, Tregs do a better job compared to conventional T cells. And this is from the monocyte surface. It is more modest. If you look at the scale, it's a little bit different with the capture from the monocytes, but still we see the same thing, that Tregs acquire more antigen compared to the T cells. And now I will quickly get into the new tools for deciphering the specificity and biology of human Tregs. And I hope I have been able to convince you about the unique potential of the antigen-specific Tregs. So it is time to really utilize the information that we already have. So one of the things is, so it is nice to use a known TCR and known antigen specificity in a well-controlled system, and to ask some biology-related questions. But humans, it is not easy to get your hands into bona fide antigen-reactive Tregs. So we have some crazy attempts right now, and I wouldn't call it crazy. So hopefully it'll give us some decent direction for our future studies. So what we try to do right now is to obtain patient PBMCs. One of the things we would like to limit is their HLA restriction right now, and I'll tell you why. I'll get into that. So the only thing that we control here right now is the HLA-DR4 allele, and what we aim to do is, by getting the PBMCs from these patients, we would like to devise some culture systems to enrich tissue-reactive Tregs. And we don't have any data right now to be able to tell you further about it, but we are working on the ways to add antigen or pancreas lysate into these PBMCs, and using some markers that are quickly upregulated in the Tregs. And these are TNF receptor family members. Okay, the other thing is, which right now Todd is doing in conjunction with their NPOD studies. They do single cell RNA sequencing with paired TCR alpha beta sequencing. which I am also after. So one of the things is, it is a great tool, single cell RNA-seq and TCR alpha beta sequencing. These are great tools, and they give you, you know, highly detailed information. But it's a matter of how to use that information from now on. So I decided to work on these information from an antigen discovery perspective. So Todd and I have already started spotting Treg and helper T cell clones from some of the NPOD sequences, so we can get our hands on the oligoclonally expanding Tregs and T effector cells. So this is basically, you can say that these are the clones that are selected in an antigen pressure, and they have expanded in vivo in these patients. So I have another magnificent collaborator, Guideng. He is also an assistant professor. He has his lab in China. He has 20 people working with him right now, so I'm really jealous with him. So he has developed a cell-based antigen discovery method, which we would like to use to decipher the antigens of these clones. So very briefly, what he does is he creates a peptide MHC2 single chain trimer library in K562 cells. So these are the artificial antigen presenting cells. And our job is to shortlist some of the peptide sequences by transducing JORCAT T cells with these human TCRs. And the next step will be co-culturing these the peptide library carrying K562s with the JORCAT T cells that carry our TCR of interest. And then I will tell you about the readout. The readout would be a highly specific trogocytosis, which is when the cells are co-cultured. K562 cell and the specific antigen-specific cell is co-cultured. Membrane parts are transferred into the K562 cells in a highly antigen-specific manner. And this is from Guideng's postdoc paper, which describes the method. So here, it's I would like to show you how dramatic it is. So they limited the antigen-presenting cell that carries the right peptide MHC2 in 1 in 10,000 dilution. And when he co-cultured irrelevant JORCAT T cell versus the specific JORCAT T cell, he noticed that trogocytosis happened only in when there's a complete antigen match. And this cell, so these K562s that carry the right peptide MHC2 are labeled with ZS screen, so they inherently have ZS screen. And you can gate on that population, and you can see the antigen-specific capture can be used as a highly specific and reliable method for antigen discovery. So what happens is that you need to sort on this population and do a NGS sequencing, and this would give you the sequence of the peptide MHC2. And we have already tested this system, whether this works for our GAD peptide and in conjunction with K562 alongside the high affinity TCR that recognizes GAD. We have seen that the high affinity TCR really passes the test, and the trogocytosis, so this is negative control, this is the trogocytosis antigen specific control. So you can distinguish the trogocytosis that is happening between high affinity TCR and the peptide MHC2. And for the low affinities, it's still a work in progress, and we are still working on to improve our detection ability for the low affinity. And I will tell you now what we are working on now. This was rather the alpha testing, the known TCR R164 TCR, and we are now curious to see if this would work or this would be of use in the real TCRs derived from real patients. And unfortunately, these patients are deceased because these sequences were derived from NPOD. But one of the good thing about these sequences is that they are derived from the end organ, the draining lymph node and the pancreas tissue. And because we have the single cell RNA-seq data alongside the TCR alpha beta information, we can distinguish which sequences come from Tregs, which sequences come from conventional T cells. And the first attempt we have is right now, okay, because these patients' information is accessible. We know that they were autoantibody. They had autoantibodies for GAD, IA2, you know, bunch of pancreas-related, autoimmune-related antigens. And some of these patients are recent onset, some of them are not so recent onsets, but they have the same autoantibody profile. So the first attempt that we are taking right now is creating our JORCAT cells that has NFAT reporter luciferase, and these are going to be transduced with patients' Treg TCRs and conventional T cell TCRs, and these will be fully functional reporter JORCAT T cells. And on the other side of the co-culture, in order to obtain antigen presenting cells, we will use T-depleted PBMCs that are derived from HLA-DR4 patients. And what we will do is we will treat them with whole proteins initially to be able to see whether there's any indication to use the autoantibody information, whether these autoantibodies are reflective of anything that may reflect the specificity of the Tregs and conventional T cells. But these are not the only candidates that we will try. So here we are trying to help our efforts to do the trogocytosis-based antigen discovery in terms of shortlisting the proteins, because proteins have to be turned into peptides. So proteins can give rise to a plethora of peptides, and our aim will be obtaining creating our single chain trimer that has the peptide sequences derived from proteins, these GAD or IA2 and so on and so forth, proteins one by one. And the aim is to create the library, this DR4 specific SCT library, using this information, protein information, and then co-culturing the JORCAT cells. And remember, this is a highly sensitive assay, which would give us a readout that is highly specific, so you can measure and sort on the trogocytosis positive cells, so the gating on the K562 cells. You can sort them based on their ability to capture the TCR, the JORCAT T cell TCR. And what we will do is, once we sort these cells, we are going to identify the single chain trimer sequence by NGS, and then we will again go back to our JORCAT cells and validate our antigenic epitope. And I am going a little bit over time, but I apologize.
No, don't apologize. This is fine. We had a little bit of a late start, and this is incredible work, really fascinating.
Thank you so much, Monica. And I am really happy to have your feedback on that. But I would like to finish by telling you another new tool that we are generating to be able to tap into the biology of antigen specific Tregs and conventional T cells. So it is nice that, you know, we are hoping to derive some antigen information out of this peptide discovery platform, and using, you know, Todd's cells that has TCR Treg avatars. But once we get into the human realm, which has multiple peptides, multiple clones, and if we can shortlist some of the candidates, what we can do is we can investigate the biology, and it's very much, it goes into the validation of the information we get from the peptide discovery. We need to find ways to validate and look into the biological relevance of the sequences, peptide sequences and TCR sequences we are getting. So I have developed a method where I aim to label the antigenic peptides, any peptide, with a small 6 amino acid tag called tetracysteine tag. And this is a profluorescent tag. So on its own, when added to the peptide, it doesn't it doesn't produce any fluorescence. However, when you treat it with a chemical compound, you can turn on its fluorescence. And right now I am finalizing a manuscript for submission towards the end of this month, hopefully. And we have gone great lengths to understand if this is a viable method that can be used to understand the biology of the T cells and Tregs that we derive from patients, and the sequences, peptide sequences that we derive from patients again. So this is just, I'm showing this to you as a proof of principle, but it may look complicated, but I'll just go slow. So here we try to understand if we can first visualize these labeled tagged peptides, and I added this so this OVA flash is the antigen that we created. So OVA is our go-to model antigen, and what we did is I derived T cells from OT2 mice and polyclonal T cells from black 6 mice. So these naive T cells, we converted them into Tregs. We did this with naive T cells too, but because I'm talking about Tregs, I would like to show you Tregs. We developed them into Tregs. So this is the OT2 Treg, this is polyclonal Treg, labeled them with different trackers, mixed them 1-to-1, and injected them into a host mouse. And we obtained our dendritic cells from a colorful mouse. This is a actin DS red mouse. All the cells are red. And the dendritic cells obtained from these mice are inherently red. We loaded them with OVA CACA. So this is the modified peptide, treated them with flash and turned on fluorescence and gave them through the foot pad. And now we're looking at the popliteal lymph node of the animal. And I would like to show you a cool video. So here you're seeing a red dendritic cell that presents the antigen that we labeled, and this is the modified antigen. We have a lot of data showing that this doesn't alter the properties of the native peptides dramatically. So okay, so what we see is these Tregs, blue Tregs, are the OT2 Tregs. And you also see, I would like to show it to you again. Okay, so what we saw here was there was an antigen-specific interaction between the Treg and the dendritic cell. Dendritic cell presents this green antigen, and this Treg that is antigen-specific comes and captures some of this antigen and carries it away with it. And this is just another way to look at the peptide MHC2 capture in action, and I was quite excited to see this. And this is a polyclonal Treg that is the control here. It doesn't carry any peptide MHC2 with it, and it doesn't really interact with the dendritic cells either. Okay, so sorry about the overtime. I would like to just summarize our findings by saying that understanding Treg biology and specificity are really key elements for successful antigen-targeted therapies. And the new platforms that we are working on and other people working on can provide us valuable information on how to modify immune networks of type 1 diabetes patients with precision. This can be by cellular biotherapy. This can be by tolerogenic peptide delivery. So it's just, it's a matter of deciding which way to go, and I am being optimistic here. And these strategies can also be used to delineate how tumor infiltrating Tregs mold tumor infiltrating lymphocyte function and repertoire. And this is also a work in progress. And please reach out if you are working on AI and machine learning based epitope prediction algorithms. I would be happy to collaborate on shortlisting some of the candidates that we can test in our downstream antigen discovery. And with that, I would like to thank you first. And here is my lab. This is my partner in crime. So my husband is a B-cell biologist. He runs his own lab. I run my own lab, but we run the labs unofficially together. So we have a nice booming research group. And I would like to thank my collaborators, especially University of Florida collaborators for the research I presented here, especially Todd, his former postdoc Benel, and his current members, Matthew, Liana, Tinzar, and my other collaborators in other institutions and in my home institution. With that, I would be happy to take any questions.
Thank you so much, Billur. This is really amazing work. You've really just done an incredible job of drilling down into these interactions and in multiple different protocol approaches, and it's beautiful. I'd like to open it up for questions. You can unmute yourself or raise your hand to unmute yourself, add a question into the chat.
Well, I am happy to answer any questions if you email me. So please feel free to reach out to me. And there is my lab website. You can follow us on Twitter, or you can message me any way you feel comfortable.
That sounds great. I think well, this will be shown on our website, so you'll have a much wider and larger audience, and I encourage people to reach out to you in that way. Additionally, to if anyone is involved in the AI/ML based epitope predictive algo generation, like you requested in the earlier slide, that would be a great person to reach out. And I don't know if you're accepting any postdocs now, but if you do, or if you are a postdoc looking, this looks like a phenomenal lab to join, with just so much work and so much collaborative effort.
Reach out to me. So yeah, we have endless possibilities here. And as I said, we are on the right now upward trajectory to discover things. So it may be a good time to join our lab.
Absolutely. This is this is a really amazing talk. Thank you. As someone has said, could you please explain a little bit about your work with MS?
That's a very good question. This is also a work in progress, and I would like to say that I am, to be honest, I am disease agnostic. I am a T-cell biologist, and my particular interest happens to be right now antigen-specific Tregs. So antigen-specific Tregs relate to a lot of autoimmune conditions. Type 1 diabetes is one of them. MS is another one of them. So now that I am in neurology department, I have access to patient samples here, and these are precious patient samples. So these are samples from CSF of MS patients as well as healthy controls. So we would like to understand what we are capable of using the type 1 diabetes system, and then this will be applied to the MS-related antigens, myelin-derived antigens, and other antigens that are implicated in the pathogenesis of MS. But we haven't started that yet. So hopefully we will derive more information from type 1 diabetes research that we have right now and funnel that information into our MS research.
Okay, well, people asked whether video will be available, and I said yes, it will be available shortly after editing on our website, www.sugarscience.org. Thank you again, Billur. This is an amazing platform you've developed and are developing. I cannot wait to see sort of where this goes. We'll check in again, probably at the end of 2023. And I think I really, it looks, you have all wheels going. It looks like you're going to have some really great results, at least even by that time. So we'll check in again then. And thank you again for this phenomenal presentation.
Thanks very much for having me, Monica. It's my pleasure.
Have a great rest of your day. Bye-bye.
Bye-bye.